Welcome back, guys. So let's do some DNA and protein synthesis. Alright, worthy of merit here is a conversation on the people that deciphered the structure of DNA. And in class, I would do like a 15-minute conversation on this. I'm just not going to do it now. I would invite you, if you ever wanted to, there's actually a movie on the subject about Watson and Crick and some of the interactions therein. That you can go in and watch. I think it's probably from the 80s. It's a neat film. I, however, will say the following. The real stars of the show, in terms of the people whom discover the nature of DNA, are Watson, Crick, Wilkins, and Franklin. Okay, Watson, Crick, Wilkins, and Franklin. Maurice Wilkins was really the guy. He ran the big lab that sort of funded everything that needed to be done to discover the nature of DNA. He brought in Rosalind Franklin as somewhat of an underling, and she worked in his lab doing what's called X-ray chromatography that builds these images of very small molecules. And long story short, she was extremely good at this, but they had a big falling out. They did not get along in any way, shape, or form. So she packed up and left, and he's sitting on all these X-ray chromatography images, and he goes to these mathematicians, in essence, Watson and Crick, and says, hey, I have these X-ray chromatography images. Can you guys make heads nor tails of it? So they built a bunch of mathematical models, and using the X-ray chromatography images, were able to work out that DNA was in fact a double helix. They built the model, and really, Watson and Crick, because they built the model, get all the glory. But there was a Nobel Prize bestowed. Unfortunately, Rosalind Franklin died very young, shortly after all this started, actually. And because of this, Wilkins, Watson, and Crick get a Nobel Prize, which is pretty fascinating. So this is a big deal in science. And let's go here. So let's talk about the basic DNA structure and how it works. Okay, so DNA stands for deoxyribose nucleic acid. It is made up of individual subunits referred to as nucleotides. These would be the building blocks of DNA. Nucleotides always have a sugar phosphate backbone and then a nitrogenous base paired unit. And we'll talk more about this in time. Yeah, here we go. So a sugar, a phosphate, and a nitrogenous base. Now, in the realm of nitrogenous bases, it is factual that there are purines and pyrimidines. The pyrimidines are small, single-ring structures, whereas the purines are much larger, double-ring structures. Alright, let's see. Let's go through and do our nitrogenous bases. So here are the purines, being adenine and guanine. Purines are very large, double-ringed. Pyrimidines are smaller, single-ringed. These are cytosine, thymine, and uracil. So purines, adenine and guanine, pyrimidines, cytosine, thymine, and uracil. Alright, so you could go down here and you can see percentages of these base pairs that are found in different organisms, and it's kind of neat to look at. And what I want you to focus on is that if your genetic code has 30%, well, hang on, I really need to give you a little bit more information. If I were you at this moment, I would write down that in DNA, all you have is adenine, guanine, cytosine, and thymine. Uracil is only found in RNA. Alright? Adenine, guanine, cytosine, thymine, that is DNA. Uracil replaces thymine in RNA. So we have these percentages here, and if you look, they kind of match up to one another. And the reason for this is because adenine only binds to thymine. So if you're going to have 27% adenine in your tissues, then you're going to have 27% thymine because they bind to one another. You can't find one without the other. It's like me and my wife. And then guanine only binds to cytosine. So if you're going to have 22% guanine, you're going to have 22% cytosine. Yeah. This is very important. You need to be able to explain that to me on exam. This matters. You can see how it kind of lines up. And these are just kind of neat percentages. I just find them fascinating. Moving on. Alright, DNA structure and function. Where do we want to go here? Let me just make some points. So what you can see here is the double helix of DNA, and then you can see if you unwind it a little bit, sort of what it looks like. This is what we have here. What we have are covalent bonds along this backbone, and in between you have our adenine and thymine, guanosine, cytosine, thymine, adenine, guanosine, cytosine, all of this fun stuff. What this is hydrogen binding together. So covalent on the backbone, this is a phosphate sugar, phosphate sugar, phosphate sugar backbone. The same thing I was talking about here, sugar phosphate. Alright, sugar phosphate. The backbone is sugar phosphate, and that is covalently bonded and very, very strong. Whereas the central portion of this, where these nitrogenous bases are, this is hydrogen binding, and it's much weaker by comparison. So basically what this lends to is when you want to divide DNA and make more copies for mitosis or meiosis, it's pretty easy to come in here and pop this apart and then make new strands using the original ones as a template. Alright? So like I could pull this piece off, and if that's cytosine, I know that it has to bind to guanine, so I can start making a new strand based off of this. And you'll see it as time progresses. Know who binds to who, which is basically what we're just talking about. Thymine, adenine, guanine, cytosine. It's referred to as complementary base pairs. This is facts. Now, on your test, what I'll probably do is drop some base sequences to you, and you'll have to tell me who binds to who. So you'll have to tell me that adenine will bind to thymine, guanine will bind to cytosine, adenine will bind to thymine, if that makes sense. And then by comparison, I might ask you to tell me what the RNA base pairs would be. And again, in RNA, we replace thymine with uracil. So adenine, instead of binding to thymine, adenine would bind to uracil. Or maybe on your test, I'll say, you know, here are two base sequences. Tell me which one's DNA and which one's RNA. Well, this one's DNA because it has thymine. This one is RNA because it contains uracil. So that's the sort of thing that I expect you to be able to do on your test. Alright, chromatin and chromosomes. This is an important chart. So what we have, you can see quite clearly this DNA double helix. So when you think about DNA, you think about this double helix, this wound, double-sided, ladder-like structure. The DNA double helix comes down. In reality, in your cells, that double helix wraps itself around very special proteins called histone proteins. Alright, histone proteins. I think I've mentioned histones to you before when we were talking about the S phase of mitosis. So in a mitotically active cell, when the cell goes through the S phase of interphase, yeah, let me make sure we're clear on that. This is a phase of interphase. When you go through the S phase of interphase, you're synthesizing new DNA and you're synthesizing new histone proteins to wrap up that DNA. So you can see the histones here. What they do is they help to package the DNA, and then you further wind and wind and wind, and you eventually get with these what we call X-shaped chromosomes prior to mitosis. When most people think about chromosomes, this is the kind of thing they think of, but it is made up of DNA in the form of a double helix. Now, let me just lay some facts on you here. Your nuclei of your cells, they don't have tightly wound chromosomes like this most of the time. Most of the times the DNA in your cells is in the form of chromatin. Chromatin is like the double helix, but not all wound up like this. It's just sort of dispersed. It's like if you took down your Christmas lights, you just make a big ball and drop them in a box, as opposed to winding them up and making them easily accessed. Right, this big mass of DNA in your nuclei, that is chromatin. Chromatin being made up of some DNA, mostly histones actually, and then a little bit of RNA that's being made at the time. So, take-home message here. The only time you see nice tight chromosomes like this is just prior to mitosis. Histones help to wrap up the DNA, and normally your DNA is in the form of chromatin, which is sort of just dispersed and all over the place. Yeah, that's perfect. Okay. Oh my gosh. You know, look at that handsome devil right there. My goodness gracious. He is so big now, too. He's like a full-grown adult. 3 years old. Alright, let's see. Basic genetic concepts. All life on Earth contains those 4 base pairs. Adenine, guanine, cytosine, and thymine. All living things have these same base pairs. Everything on the planet. They all code for biological parts. Let's see. I've got some genetic similarity chart here. It's fascinating. You can see that like the proteins we make, all mammals are very similar to one another, which should make sense to you. We're all from the same genetic lineage, so we can all make pretty much the same proteins. And our genetic code's quite similar as well. You've probably heard people say that we are quite similar to chimpanzees. That is a fact. Okay, they contain about 98% of the same genes as we contain. They are very similar to us genetically. We as humans, as a single species, share about 99.9% identical genetics. So think about the two most different people you've ever met. They contain 99.9% identical genetics. There's very little genetic variability in reality. Like all the differences we see in people are just phenotypic. We express things slightly different. Yeah, so we're quite similar. We are quite similar. Quite similar. What do we want to say here? So there are some fascinating things that can happen with genetics. This stuff that's kind of cool to me. So my point here is, me and my red hair, and then my son's got my red hair as well, which is great for him, I suppose. This is from genetics. So the reason we look similar is all based off of our genetic code. Okay, minor little tweaks in our genetic code, which leads to us having a similar appearance because we are related to one another. An example of this is a mutated MC1R gene, which I have and he does too. That's what causes red hair. It leads to some other kind of neat variations. So my take-home message of this slide is that we are all very similar to one another, but little minor tweaks in our genetic code can result in some kind of fascinating augmentations. Okay, it's good to have some variability in genetics. Let me just expand on that a little bit. And I probably mentioned this multiple times throughout the semester. So the more different the genetics of two individuals are when they have offspring, the more likely those offspring are going to be healthy. Where you run into problems with genetic disease is where you have two individuals that are quite closely related having offspring. And I probably did this whenever we talked about inbreeding depression in the European royal families. The idea is that if you breed very similar organisms together, they are at an increased risk of having what we call, oh dudes, I'm totally getting deep here with you, the accrual of deleterious alleles. That's a fancy way of saying two people that are quite similar, if they were to have offspring, that would increase the chance of that offspring getting two bad copies of a gene and then having a genetic disease. Yeah. So having a lot of genetic diversity in a population increases what we call the genetic fitness of that population, which leads them to be more healthy overall. So I guess what I'm telling you is if you're contemplating having children, try to have children with someone very different from you, and that's going to vastly increase their chance of being genetically healthy. That's a weird statement. It's actually, there's a term for it, a scientific term. It's called hybrid vigor. Okay, hybrid vigor. Anyway, moving on. That's why my little dog, whom is, pardon the terminology, but a mutt, if you will. So he is rescue and he's perfect. His parents were probably very genetically different from one another, and as a result of that, he's like 12 years old and totally healthy, as opposed to purebred dogs, whom tend to die very young from a lot of genetic problems. Okay, now then, let's move on. So, boy, let me tell you, all of this is important. I want you to know every bit of it. I kid you not, I want you to know every bit of it. So, what we have here is our DNA double helix, and our DNA double helix, and these are the, let's see, DNA polymerase, ASE, what are they? They are enzymes. These are the enzymes that lead to DNA replicating. Alright, now we call DNA replication semi-conservative. Let's see, let me just explain what that means, semi-conservative. The idea is that this is semi-conservative replication because when you take one DNA strand and you make two DNA strands out of it, the two new strands have a piece of the original. Let me say that again. Here is the original double-stranded DNA double helix. When you go through mitosis to make two new copies of that initial strand, the new ones, the new copies, have a piece of the old initial strand. So it's semi-conservative because the new pieces have to be made on the old piece. It's a fancy way of saying that it's very hard to screw up mitosis. It's very hard to screw up replicating your DNA. Your DNA is replicated with extremely high accuracy, and that's why we tend to see a lack of mutations as a result of this. All right, let's talk about these enzymes. So we have DNA helicase. DNA helicase unwinds the DNA double helix. We have DNA polymerase. DNA builds the new strands of DNA. It's kind of a fancy way to say it, but I'll get there in just a second. And then we have DNA ligase, and I'll explain that in a moment. Basically, it fuses together these structures called Okazaki fragments. Yeah, and I'm just not going to worry about binding protein at this particular moment. So let me see if I can lay this out for you. DNA helicase comes in. Let me make sure I got time for it. Yeah. DNA helicase comes in, and it splits open the DNA double helix. Okay, what the helicase does is it cracks it open. All right, helicase binds to and unzips the zipper of the DNA double helix. And what that does is it frees the two individual strands so that they can then be worked on and replicated. Now, if helicase unwinds it and cracks it open, polymerase is what comes in here and makes the new pieces. There's a polymerase and there's a polymerase. But we have a mild problem, all right? This is a weird one, so prepare yourselves. DNA polymerase can only read one direction. And believe it or not, your DNA is very much directional. It only reads, for instance, this strand, based on this image, reads this direction, whereas the other strand would have to be like its mirror image almost, so it has to read the opposing direction. So this one reads that way, this one reads that way. It's just how this works. Don't think about the deeper details of this. Just understand that DNA is directional. In one direction it'll read that way, and then the other one it'll read the opposing direction. So the side effect of this is that DNA polymerase on the initial, what we call a leading strand, it can just zip straight down the length of that leading strand and make a brand new piece of DNA, man. It can just go straight down the length, making a new piece of DNA. All is well in the world. It's totally fine. The opposing strand, however, it's more complicated. All right? What it has to do is wait for a section of DNA to be unzipped, and then it'll attach and zip down until it reaches where it stopped previously. Then a new piece of DNA opens up, and it'll attach and zip down to where it stopped previously. So what you end up with are all of these little fragments. All these little fragments of DNA that we call Okazaki fragments. All right? They're called, it's a Japanese person that discovered them, if I'm not mistaken. They make what are called Okazaki fragments. And these Okazaki fragments have to be fused together. They have to be bound to one another. And that's done by DNA ligase. All right. So DNA helicase unwinds it. DNA polymerase makes the new pieces by laying down new base pairs. And then we have DNA ligase, which mends together the Okazaki fragments on the what's called the lagging strand. All right, the lagging strand. And you can see that here. So what we have, let me just go back and forward. We have opened up helicase. We have a piece of DNA just being made by DNA polymerase as it goes down. And then the opposing side, we're getting all these Okazaki fragments that have to then be mended by DNA ligase. Yeah, that's perfect. That'll do. Here's some more Okazaki fragments. You can see this on the opposing side. Yeah, hopefully that helps. Okay. Replication errors. Let's see, we have time. Let's restart. Replication errors. So DNA replication is super accurate. Super accurate. The idea is that DNA polymerase, it's hard for it to screw up just based off the way it's set up. Like, you have very specific base pairs that can only bind to other base pairs very specifically. So the chance of DNA polymerase laying down those base pairs wrong is very limited, very rare. And then further, DNA polymerase tends to go back through a second time and proofread its own work. So if it finds any errors, it can actually fix the errors as it goes. And that's great. That's great. So it's very much capable of fixing any problems that it finds. This really limits the chance of mutation in our tissues. And that's a good thing. Gene expression. Okay, so let me just read to you a little bit. The flow of genetic information is from DNA to RNA to proteins to observed traits. So, we contain DNA. That DNA is the code for how to make a protein. All right? How do you get from a DNA to a protein? You do that through an intermediary we call RNA. RNA takes a copy of a gene in your genetic code and carries it out into the rough endoplasmic reticulum where proteins get made. And the proteins that we make lead to the genetic traits we display, like red hair or my daughter looking just like me. And what a cutie she is, all right? Yeah, so we got to go through and talk about this as a mechanism of gene expression. Let's see, what all do I want to say here, folks? Yep, I've pretty much already said all this stuff. Through protein synthesis, genes are expressed. DNA in a gene determines the sequence of nucleotides in an RNA molecule. So DNA is the code, and RNA is a copy of that code. And RNA leads to the functioning of or leads to the making of a protein. Yeah, I've already said all that. All right, so let's talk about it. Now, here is some broad, open statements that you need to be familiar with. Your DNA contains many chromosomes. Your DNA contains many chromosomes. 23 pairs of chromosomes if you want to get technical. Chromosomes have many genes. All of your genetics, you have about 25,000 genes total. That will be areas that code for a protein. Now, do all of us have the exact same genes? No, we don't even remotely look identical. Variants of genes are called alleles. So, maybe you'll have alleles for blue eyes. Maybe you'll have alleles for green eyes. Maybe you'll have alleles for brown eyes. It's all for eye color, but it is different variants of that gene. Those are alleles. And let's see, how gene activity... Yeah. What's the point? The point is to make proteins. Now, proteins are very, very important for us. They are made out of amino acids, of which there's only about 20, and they have a variety of functions. So proteins can be supportive. Let me make sure I'm not going to write on my screen here. Proteins can be supportive like keratin. Keratin is the protein that makes your skin tough, which is good. So any blunt force trauma doesn't just slice your skin right off. No. Keratin makes your skin very tough. Enzymes like amylase. Amylase helps us break down starch in our intestinal tract, which is very important for us. We have to be able to break down foods in order to absorb them. Transport like hemoglobin. Hemoglobin is the iron-containing red molecule that's found in your bloodstream. So the reason your red blood cells are red is because they contain hemoglobin, and hemoglobin helps us transport oxygen. Like, we can play this game all day long. You know how important insulin is as a hormone. This is a protein. Actin and myosin is what allow your muscles to flex. This is all very important, all right? All very, very important and meaningful. So the idea is, and I've said this to you before, but every time I look at you, everything I see is protein. Your hair, your skin, your eyes, your nails, all of it. It's all protein. So proteins are very important for us. Cells certainly have a molecular chain of command. So DNA is in the nucleus and it can't get out. And you know why? Because DNA as a molecule is just too big. DNA as a molecule is too large to escape the nucleus. So what we have to have is a small copy of a gene that we want to use to make proteins. That copy of a gene is what we call RNA. Okay? DNA contains lots of genes. RNA would be a copy of a single gene that you need. So let's say that your blood sugar levels rise. Then that would mean that you need insulin to tell your cells to bring your blood sugar levels back down. So your DNA will copy a piece, make an RNA strand, for how to build insulin. And then it'll offload that out to the rough endoplasmic reticulum where proteins are made. Now we apply terms to this. These terms are transcription and translation. Transcription and translation. Transcription is taking DNA and making RNA. Translation is taking RNA and making protein. Now these have... sometimes you just got to have old Daenerys Targaryen in your image. RNA is a single-stranded unit. Always, RNA will be single-stranded. It's very simple. Whereas DNA is a double helix. Point to be made. Sometimes when you type in on Google and you say, show me a picture of DNA, this pops up and you just have to include it. Alright, let's talk about the differences between DNA and RNA. DNA is always found inside the nucleus because DNA is too large to escape the nucleus. RNA is made in the nucleus, but it does its work out in the cytoplasm of the cell. DNA replicates for cell division. RNA is going to be made when proteins are necessary. DNA is made of adenine, guanine, cytosine, and thymine. RNA is made of adenine, guanine, cytosine, and uracil. Deoxyribose is the sugar in DNA. Ribose is the sugar in RNA. DNA is always double-stranded. RNA is always single-stranded. Yeah, okay. Okay, so let's go here. So transcription and translation. What I need you to know is that transcription happens in the nucleus. So we have our DNA strand. We're going to make a little copy of a piece of one. That is called transcription. That can then leave the nucleus, go out into the cytoplasm, where translation occurs. That would be taking that RNA strand and making a protein from it. That typically happens on the rough endoplasmic reticulum. And here I tell you that I have, at least to some degree, been lying to you. Okay? There are, in fact, 3 different types of RNA. And those 3 types of RNA are messenger RNA, transfer RNA, and ribosomal RNA. All of these are different from one another. They all contain uracil instead of thymine. They are all single-stranded, and they all are very important. Okay, so let's talk about it. I can guarantee you, you're going to be seeing this on your exam. What I love to do is put, like A is messenger RNA, B is transfer RNA, and C is ribosomal RNA, and then I'll go through and give you like 10 scenarios, and you'll have to tell me which one's which. Alright, let's roll. So, first comment, I suppose. Have I talked about RNA polymerase at all? I don't think I have. Let me tell you something real quick. This is very important as well. So you realize at this stage that DNA polymerase is what's used to make new strands of DNA. Okay? DNA polymerase is used to make new strands of DNA. Well, there's also an enzyme called RNA polymerase. RNA polymerase makes messenger RNA. Let me say that one more time. RNA polymerase makes messenger RNA. It cracks open the DNA molecule. Here's an RNA polymerase molecule. DNA is cracked open, and the RNA polymerase will come in, find the gene, and make a copy of that gene, and then kick off. The copy of a gene from DNA is called messenger RNA. Let me say that one more time. When we come into the DNA and we make a copy of a gene, that copy of a gene is called messenger RNA. It's called messenger RNA because it carries a message. Okay? That is going to go out and then be translated. The product of transcription is messenger RNA. That is it, right there. RNA transcript. That is messenger RNA. Transfer RNA is different. Transfer RNA is kind of neat. It's got 2 binding sites. Transfer RNA molecules will have a binding site called an anticodon site, and then a secondary binding site called an amino acid binding site. What transfer RNA does is it carries amino acids around. What do we make proteins out of? We make proteins out of amino acids. How do those amino acids get to where they're going to make protein? They're carried by transfer RNA. Transfer RNA carries amino acids to this piece to make proteins. Okay? Bear with me, I'm going to talk more about this. Very important. Transfer RNA has an amino acid binding site and this anticodon site. And then last but not least, we have ribosomal RNA. Ribosomal RNA, this is basically just a ribosome. Ribosomes are 2-part systems, a top half and a bottom half, and they clamp together on a messenger RNA strand and zip across it like a zipper, making proteins. So ribosomes make protein. Let me say it again. Ribosomes make protein. The reason we call rough endoplasmic reticulum is because it's studded with ribosomes. And those ribosomes are actually made of RNA. Okay? They're made of RNA. So ribosomes are basically pieces of RNA. And the whole thing together, hang on, is going to look like this. Now, what you have here is a top and bottom half. This green and this yellow, this is a ribosome. This strand that's coming in here, all right, that strand is messenger RNA. So you can see the top and bottom half coming together, clamping down on the messenger RNA strand. And you can see these little blue things come flying in here. These are transfer RNA molecules. So you have the ribosome, that will be ribosomal RNA. You have messenger RNA here as a strand, and then you have these transfer RNA molecules coming in and bringing amino acids. And every time they zip through, they're laying those amino acids in and covalently bonding them together, and they're making a primary strand of a protein. So how do you make a protein? You have a ribosomal RNA piece coming together, you have a messenger RNA piece, and then you have these transfer RNAs coming in, leading to the formation of that protein. Now, let me say this as well. You don't just have one transfer RNA I'm sorry, let me try again. You don't just have one ribosome on one RNA strand, messenger RNA. That's not how this works. The reality is what we call a polyribosome, and you can see that here. Basically, if there's a messenger RNA strand hanging out in the rough endoplasmic reticulum, it'll be bound to every ribosome available at any moment. So that thing's going to be completely packed with ribosomes, and they're going to be making as much protein as they can physically make in the time that's allotted for them. Yeah, okay, that'll work. Final thought. So this process is incredibly fast. It's incredibly accurate. We can measure it. Bacteria are capable of laying down amino acids at a rate of about 20 per second, and only 1 in 10,000 is incorporated incorrectly. So your chance of having a mutation is very, very limited. And further, this is an energy requiring process, so it's going to take a lot of ATP to make all of these steps go. Transcription, translation, all of those are energy requiring. So it uses a lot of cellular energy, a lot of ATP. The role of genes. Okay, so information in your genes is ultimately going to be used for making proteins. The DNA in your genes contains information for... let's just discuss. Here's the take-home message. I can hear my kids over. They're driving me crazy. Alright, so this is what happens. You have 23 pairs of chromosomes. Those 23 pairs of chromosomes contain genes. About 25,000 genes total. Each gene is the code for how to make a single protein, right? And you can make a copy of that gene. That would be a messenger RNA strand. They can leave the nucleus, go out into the rough endoplasmic reticulum, where it can find a ribosome, and that ribosome will translate a protein using transfer RNA molecules to bring in amino acids from that initial, what we call RNA transcript. Yeah, and that's really what this says, but just in a better format. I don't think that I want to get too deeply into this, so... Hang on, let me go check on my kids, folks. I'll be right back. They're going crazy. Hi, guys. In order to save lives here at the house, we're hanging out together for a few moments. This is where it all gets real for us. So the reason I point this out, this codon system, is because your transfer RNA molecules, they're actually set up with 1, 2, 3 amino acids, and they'll only bind to certain spots on an RNA strand. Okay, they'll only bind to very specific places. The essence of this is, and I'm not going to make this real complicated. I'm not going to test you on it with intensity. The idea is that because of this 3-part system, these codons of DNA, so this is a DNA codon, or actually these are RNA codons, RNA codon, RNA codon. Because of this system, your capacity for laying down amino acids is incredibly accurate. So your transfer RNA molecules, they're just incredibly accurate. The take-home message to this is that the codon system leads to your transcription and translation being very, very accurate. I know. So you have very decreased risk of mutations. Okay, good enough. Decreased risk of mutations. Alright, now a couple of points to be made. There are what are called start codons and stop codons on every... I know. Start codons... This is how you keep anybody from getting too upset in the house. We have what are called start codons and stop codons on all of our genes. So in your genetic code, if you can look through it, there would be like an AUG that'd be adenine uracil guanine on a DNA or an RNA strand, for example, and that would basically say this is the beginning of a gene. Okay, this is the beginning of a gene. Or alternatively, there are stop codons that say, hey, this is where the gene stops. So this is where, I know. This is where it starts, and this is where it stops. So this is a handy way of making sure that our messenger RNA strands aren't made up of multiple genes, because they can, I know. Well, let's see if I can go and do something else to this kid. I'll catch up with you in a second. Alright, let's see if we can finish this thing out. So, I just have just a couple more slides on these concepts. Okay, so DNA, when we make an RNA transcript of it, that messenger RNA strand, from the start codon to the stop codon, there's actually more in there than you really need. You see, a messenger RNA strand initially is made up of what we call exons and introns. Okay, there are exons and there are introns. The exons are what we call coding regions. The way I think about it in my own brain is that exons are expressed. So the protein you're going to make, that is basically the exons, right? So exons are expressed. These are the coding regions that lead to the manufacture of a protein. Introns are areas of what we're going to call like nonsense DNA. These are non-coding regions. They don't have any use for our purposes at this particular stage in life. So what will happen is the RNA molecule will have a enzyme come in and splice out the introns. So you kick out all the introns and leave nothing behind but the exons, and that is the messenger RNA strand that leaves the nucleus and goes out into the cytoplasm for translation. So you'll transcribe the messenger RNA strand, you keep the exons, you kick out the introns, and the strand that results is going to be used for the manufacture of a protein. So you have parts of your DNA that are non-coding. That's the take-home message here. And those non-coding regions are referred to as being introns. Alright, here's a nice breakdown of everything we just talked about. You got your DNA, got your exons and introns and messenger RNA. You can see this messenger RNA strand coming out here and attaching to a ribosome, and how the ribosome uses transfer RNA molecules to make proteins. And here's the same thing again. Nucleus to rough ER to the Golgi to secretion of functional proteins. We've played this game before, and that's really as far as I want to go. There's some other neat stuff in here about splicing in genes, and it's pretty cool stuff. But it's not test material, so I'm not going to get into it, being that we are so time limited. All right, and that's the extent of Chapter 12, DNA and Protein Synthesis. I'll be putting up a quiz on this, and it'll be due in about a week after the video posts. All right, thanks, guys.